But I'm going to talk about this one here. And this what's got our attention is that SPINT was the top correlated gene in both data sets. Here's our entry in, right? I've used statistics, I used the crowd to kind of get me to this point. Now I can potentially have a target to look at. So what is SPINT? It's a serine protease inhibitor. So what it does is it stops other proteins from cutting up other proteins, which you can imagine would have a huge effect, right? How functional would you be if somebody cut off your arms? Not very functional. I guess it depends what you're doing, right? Proteins don't like to be cut. So, this is inhibiting a lot of things. One of those things is a hepatocyte growth factor that's been previously implicated in all kinds of aggressive tumors. Also, in another type of cancer, and you see this a lot, if I'm working on lung cancer and I'm looking at a gene, usually that gene has been characterized in another cancer as well. And sure enough, this gene, the SPINT1, which is positively correlated to our gene of interest, is known to suppress metastatic pulmonary colonization of pancreatic carcinoma cells. And also, I will say, again, there is no prior knowledge of this SPINT1 ever acting with jupe. So I come from the bench, and anything I tell my collaborator, I want him to validate, right? I don't believe something, it's nice to play with the numbers, but you got to prove it somehow. So that's basically what he did. And what my collaborator did, his name was Rob Wynn. He's now at University of Illinois now. He took a bunch of cell lines that some would have junction plecoglobin expression like these here, and some didn't. And what he noticed is that it's also correlated at the protein level. That in these two cell types that don't have any jupe, you either have little or no SPINT1, right? Also, when you overexpress jupe, then SPINT1 goes up in this cell type. The thing about this cell type, though, it has a functional copy of P53. Who's heard of P53 at all? All right, good. Did you guys take Jaeger's class yet? Yeah, okay. Yeah, he talks about P53. So there's one, you overexpress it, SPINT1. So you're showing that relationship there. But what they found is that this doesn't work unless you have a functional copy of P53. So here they overexpress jupe, nothing happens. Then they overexpress P53, and then SPINT1 goes up. Now we're getting a mechanism. We're having this relationship here. Now we have a mediator. Not only do we have a mediator, but we have a critical mediator because this is P53. And this is like the ultimate cop in your cell. If something goes wrong, it shuts everything down. And as we were doing this, at the time he was kind of looking at me like I was crazy. Like, there's no way you can figure all this stuff out, just with numbers. You're using two different data sets. You didn't even generate the data. I'm like, yeah, I know. That's why this is cool. That a paper came out that actually showed a relationship to junction plecoglobin. And not only that, to junction plecoglobin to P53. And here it's actually inducing this 1433. This also showed up on our list. But now what I'm saying is not only do we see this gene, but it's also inducing this SPINT1. And so now they've been playing with this as a potential therapy for metastatic tumors, right? If you take this cell line and you overexpress SPINT, the proliferation of these cells goes down, right? Even in the one that's lacking P53. This is a potential therapy that it can use. Not only does it decrease the proliferation, but it increases the migration. As you can imagine in cells, a metastatic tumor, the faster it can migrate, probably the more aggressive it is. So what they found is when they put this SPINT1 back into these cell lines, that here they didn't put it in, after 12 hours, basically what they do is they'll grow these cells on a petri dish and then make a line through it, and then they determine how fast it'll grow back together. Here it grew back in 12 hours. But now when they take that same cell line and they add that SPINT1, now this is after 24 hours, they still haven't even grown back together, right? We use the bioinformatics to find something useful. Now we can use it as a clinical potential therapy. And now he's got a mechanism. And this was actually, we published this a little while ago. And we are the first people to know this. And it was easy. That gamma continuum or junction plecoglobin induces this P53, which induces SPINT1, which probably inhibits this growth factor, which inhibits proliferation and migration. Okay, I do have time.